Hello. Today we will talk about an example of biological method. That example of biological method is study of malaria. We know that malaria is a common disease in many countries, including Pakistan. Let's talk about history of this disease. We will go through the history of this disease to know how biology solved the biological problem concerning the cause and transmission of malaria. In ancient times, more than 2,000 years ago, physicians were familiar with malaria. They described it as a disease of chills and fevers with recurring attacks. Here, the word recurring means the something that happens repeatedly. Physicians also observed that the disease was more common among people living in low, marshy areas. It was thought that the stagnant water of marshes poisoned the air, and as a result of breathing in this bad air, people got malaria. This belief led to the name of this disease. From this diagram, you can understand the belief about malaria at that time. In this diagram, you can see that stagnant water of marshes is poisoning the air, and breathing of that bad air, people got malaria. But that was just a concept about malaria at that time. What is the meaning of world malaria? World malaria IDS derived from two Italian words. These two words are mala and aria. Mala means bad, and aria means air. For further clarification of the observation, some volunteers drank stagnant water from the marshes. They did not develop malaria. In the 17th century, history of malaria was. In 17th century, when the New World, America, was discovered, many plants from America were sent back to Europe to be used as medicines. The bark of a tree of these plants, known as quina, was very suitable for curing fevers. It was so beneficial that soon it was impossible to carry enough bark to Europe. Then some dishonest merchants began to substitute the bark of another tree that is cinchona, which closely resembled quina. This dishonesty proved much valuable for mankind. Cinchona bark was found to be excellent for treating malaria. So here's the question that why cinchona bark is effective agonist malaria. Reason is that cinchona bark contains quinine that is effective in treating the disease. At that time, physicians treated malaria with cinchona without understanding the cause of malaria. Then 200 years later, it was found that some diseases are caused by tiny living organisms. These living organisms include bacteria, viruses, Plasmodium, euglena, paramecium, amoeba, etc. These living organisms can be seen by microscope. After this discovery, it also became a belief that malaria too might be caused by some microorganism. In 1878, a French army physician, Laveran, began to search for the cause of malaria. For that search, he took a small amount of blood from a malarial patient and examined it under microscope. He noticed some tiny living creatures. His discovery was not believed by other scientists. 2 years later, another physician saw the same creatures in the blood of another malarial patient. 3 years after the second discovery, the same creatures were observed for third time. The organism was given a name Plasmodium. In the last part of 19th century, many different causes of malaria were being suggested By that time, there were 4 major observations about malaria. According to biological method, second step of biological problem is observation. So according to biological method to solve this problem, 4 major observations about malaria are given as malaria and marshy areas have some relation. Quinine is an effective drug for treating malaria. Drinking the water of marshes does not cause malaria. Plasmodium is seen in the blood of malarial patients. We know that a scientist uses whatever information and observation he has and makes one or more hypotheses. Making hypothesis is the third step of biological method. The hypothesis made in this case was Plasmodium is the cause of malaria. Scientist does not know whether his hypothesis is true or not, but he accepts it may be true and makes deductions. Making deduction is the fourth step of biological method to solve a biological problem. One of the deductions from the above hypothesis was, if Plasmodium is the cause of malaria, then all person ill with malaria should have Plasmodium in their blood. The next step was experimentation, and that is the fifth step of biological method. To test the deduction through experiments which were designed as blood of 100 malarial patients was examined under microscope. For the purpose of having a control group, the blood of 100 healthy persons was also examined under microscope. After experimentation, then step is summarization of results. This experiment has the 2 results. First one is, showed that almost all malarial patients had Plasmodium in their blood. Second result is, 7 out of 100 healthy persons also had Plasmodium in their blood. This diagram is representing above results. On one side, this diagram shows that almost all malarial patients had Plasmodium in their blood, while on the other side, this diagram shows that when the blood of 100 healthy people was taken, it was indicated that blood of 93 healthy people did not contain Plasmodium, while in the blood of other 7 people, Plasmodium was present. Now we know that Plasmodium in the blood of healthy people was an incubation period. So what is incubation period? So it is the period between the exposure to an infestion and appearance of the first symptom. We can also say that it is the period between the entry of parasite and host and the appearance of symptoms. So above results prove that the hypothesis, Plasmodium is the cause of malaria, was true. So that is the result of first biological problem. Let's discuss about second biological problem by biological method. Next biological problem was to learn about how Plasmodium gets into the blood of man. Biologists were having following observations. These observations are given as, first observation is, malaria is associated with marshes. And second observation is, drinking water of marshes does not cause malaria. From these observations, it can be concluded that Plasmodium was not in the marsh water, but it must be carried by something that comes to marsh water. In 1883, a physician, A.F.A. King, listed 20 observations. Some important observations of King were that first observation was people who slept outdoors were more likely to get malaria than those who slept indoors. Below diagram, you can understand the first observation, that people who slept outdoors are more affected by malaria than the people who slept indoors. Second observation was people who slept under fine nets were less likely to get malaria than those who did not use such nets. Below diagram, you can understand that more clearly. And third observation was individuals who slept near a smoky fire usually did not get malaria. Below diagram, you can understand this observation. On the basis of these observations, hypothesis is the second step of biological method. King suggested a hypothesis. Mosquitoes transmit Plasmodium and so are involved in the spread of malaria. After making hypothesis, deductions are made. Following deductions were made considering the hypothesis as true. If mosquitoes are involved in the spread of malaria, then Plasmodium should be present in mosquitoes. A mosquito can get Plasmodium by biting a malarial patient. In order to test the above deductions, Ronald Ross, a British Army physician, working in India in 1880s, performed important experiments. He allowed a female Anopheles mosquito to bite a malarial patient. He killed the mosquito some days later and found Plasmodium multiplying in mosquito's stomach. The next logical experiment was to allow an infected mosquito having Plasmodium to bite a healthy person. If hypothesis was true, the healthy person would have got malaria. But scientists avoid using human beings for experiments when results can be so serious. Ross used sparrows and redesigned his experiments. During experiment, he allowed a female Culex mosquito to bite on the sparrows, suffering from malaria. Some of the mosquitoes were killed and studied at various times. Below diagram, you can see mosquitoes are biting malaria-affected sparrow. Ross found that Plasmodium multiplied in the wall of mosquito's stomach and then moved into mosquito's salivary glands. He kept some mosquitoes alive and allowed them to bite healthy sparrows. Ross found that the saliva of the infected mosquito contained plasmodia, plural of Plasmodium, and these entered the sparrow's blood. When he examined the blood of these previously healthy sparrows, he found many Plasmodia in it. In the end, the hypothesis was tested by direct experimentation on human beings. In 1898, Italian biologists allowed an Anopheles mosquito to bite a malarial patient. The mosquito was kept for a few days and then it was allowed to bite a healthy man. This person later became ill with malaria. In this diagram, you can see the conduction of experiment on sparrow and human. On sparrow experiment, malaria-affected sparrow is bitten by Culex mosquito. Then, when this Culex mosquito bite this healthy sparrow, then this healthy sparrow become also affected by malaria. So it is proved that mosquitoes spread malaria. On human experiment, malaria-affected human is bitten by Anopheles mosquito. Then, when this Anopheles mosquito bite this healthy human, then this healthy human become also affected by malaria. So it is proved that mosquitoes spread malaria. In this way, it was confirmed that mosquitoes transmit Plasmodium and spread malaria. If you like this video, then subscribe this channel. Thank you.